I hope you're ready to settle in for our final and arguably most powerful session of the day. We welcome a group of individuals who support or are leading CGT drug development programs. Kim Nye, caregiver, founder, and executive director of the TESS Research Foundation, will be joined by Dr. Joe Katakowski, director of research at the RTW Foundation, who provides advice and assistance to other rare disease foundations. Michelle Krueger is a caregiver and co-founder of Elly's team who worked with RTW Foundation to facilitate a CGT treatment for her daughter. Our panelists will be exploring the different processes they went through as patient advocacy groups to move a gene therapy from early research models to first in human. Again, please ask questions throughout these sessions using the Q&A feature. We will monitor these questions and answer them live. We will first begin by having each panelist briefly introduce themselves. Please, let's start with Kim.
Hi, everyone. Thank you to ASGCT for putting this panel together. I'm really excited for it. As Rachel said, I'm Kim Nye, and I'm the founder and executive director of TESS Research Foundation, where we're focused on a monogenic disease called SLC13A5 epilepsy, or also known as SLC13A5 citrate transporter disorder. It's an ultra rare disorder, and it's a neurodevelopmental disorder with the main symptom being seizures that start within hours or days of birth. Two of my four children have the disorder, and our goal at TESS Research Foundation is to develop therapeutics, so not just a single therapeutic, but multiple potential treatments in order to treat this disorder. And we're trying to develop those for our entire community. So it's not an N of one approach. And we're looking at patients all the way from birth until later in life. For instance, my children who have this disease are now 12 and 22, so significantly older. When we first received a diagnosis, my children were among the very first in the world. So there really was nothing in place for this disorder and very little known about it. Which is not the position that you want to be in as the parent. But I managed to, a few months later, a publication came out and it suggested that there was a drug that might be the cure to this disease. So the first thing I did was not start a foundation, but instead reach out to that drug company and see if that medication would work in my children. And when it did not, we really had to pivot. And so that is when we started TESS Research Foundation in 2015. And we basically decided, okay, we need an infrastructure here. We need to figure out how we're going to learn more about this disorder because clearly the initial thoughts about it were not accurate. And that's when we really put in place an infrastructure of finding more patients. We went from having just a handful of patients, mostly in my house, to having 150, diagnosed families, like kids and adults. And we also put in place, a natural history study and animal models, all these things that I think you've heard a lot about earlier today in some of the presentations. And we're really hopeful that we will have a treatment that does change the trajectory of this disease. The program that I think we'll talk the most about today is also the program that is farthest along, which is our AAV9 gene therapy, which was developed by Rachel Bailey, actually. So she knows a lot about what we're doing in this space. I guess just for everyone's information, we're based out of California, and I think that it's been really exciting to see all the progress that's happening in this space, and I'm really, I think our story really has a nice complement with Michelle's story, which is a really exciting story as well. So maybe I'll leave it at that for the introduction and hand it back to you, Rachel.
All right, thank you so much, Kim. Joe, if you'll introduce yourself next.
Sure. I'm Joe Katakowski. I'm the Director of Research at the RTW Foundation. We're located in New York City. In my former career, I worked as a preclinical R&D researcher, mostly in the biopharmaceutical industry for major pharma companies. Spent about a decade there. I bring that drug development expertise to our foundation, where my work largely focuses on supporting patient-led rare disease foundations that are trying to advance therapies, specifically genetic medicines like gene therapies, ASOs, things like that. And our main service that we provide to the rare disease community is our Rare Disease Advising Program, where we work with dozens of ultra-rare disease organizations at various stages of growth to help them navigate everything from early scientific strategy to translational development, identification of partners. And a big part of what we try to do is just help families and foundations translate urgency into credible therapeutic programs. So the Rare Disease Advising Program is free. We try to empower foundations by giving them actionable advice on the research partners to work with, prioritization efforts for what modalities might be of highest interest. And we also conduct disease landscape reviews to sort of get into the nitty gritty of the biology of the disease. And that makes our advising services more meaningful because we can speak with some authority and some confidence into the mechanisms of the disease when there is literature to support that. So we provide that to our foundation partners as well. And we've been doing this probably since we met Michelle, really. I think that's sort of where we piloted this effort and extended it from there. So we're hoping to scale this and expand it to others in the rare disease community.
All right, thank you, Joe. And that's actually an excellent segue for Michelle to now introduce herself.
Yeah, thank you so much for having me. My name is Michelle Krueger. I am most importantly the mother to Elly, who's a 2.5-year-old baby girl, and also Addie and Charlie, who are 5 and 6 years old. I'm also the founder of Elly's team and the CEO. My story is a little bit unique, and I think it's going to be really interesting to talk about our path versus Kim's path, as she alluded to. I created Elly's team after my daughter was diagnosed with an ultra-rare neurodegenerative disease called netamus, which is caused by a mutation in the IRF2-BPL gene. She was diagnosed at 10 months of age, and when we found out about it, we learned a couple unfortunate truths very quickly. The gene had just been discovered in 2018, so very, very little was known about it. There were documented only 40 to 50 kids with it. We now believe there's more like 150 or 200, and it is regressive and degenerative for a subpopulation of the kids. I remember, when we got diagnosed by Dr. Grinspan out of Weill Cornell in New York City, he said, I don't know much about this. No one knows much about this, but what I am seeing is it's really, really bad. And he said, there's no treatments, there's no cures. And many of you who are parents probably remember getting that exact news. I think the difference in my story is that Grinspan didn't stop there. He didn't say, go home and love your child. He said, but gene therapy is becoming a reality. I don't know much about it. I've never done it, but if you want to do it, I'll do it with you. And that was really the launching point of Elly's team. A couple months later, after we kind of digested the devastation of what we were told, we started Elly's team, which is a faith-based organization with a singular purpose of translating medical research to treatment in record time. We specifically picked that because we had to. My daughter was very severe. And unfortunately, a couple months after her diagnosis, we had a repeat MRI that indicated she already had brain atrophy. So at that point, we had already started on our journey, which we'll get into further. But I remember thinking, okay, I have to go fast, and I have to go even faster than I thought I had to. And so we treated Elly, with an AAV9 gene therapy on April 3rd of last year. It was 10 months after the pursuit of our gene therapy program, and 14 months after the day of her diagnosis, which I think is one of the fastest programs that has gotten to the clinic with an AAV9. There's a lot of reasons we were able to do that, which we're going to get into, but one of which is because this was an N01 and a very severe child, which has a very different pathway than what Kim is doing in her organization, for instance. Now, with that being said, Elly's team is still very focused on the broader population. We, at the same time as we were pursuing the AAV9 N01 program, we also funded projects in ASOs, drug repurposing, gene editing, and we're continuing all of that. Also, at the day we were diagnosed, there was no mouse model. There's still no mouse model, but we're working with Jack's Labs on that. There's no animal models. There's very few cell models. So we were in some ways starting from scratch. But what we were lucky enough to kind of step into is Dr. Catherine Meyer had done some preliminary research on the gene from some families that had come before us. And she had a hunch that she thought gene therapy might work. So we'll get into kind of the details of my program a little bit later, but I think I'll pause there with that intro.
All right. Thank you so much. I mean, all of you have such powerful stories, and I think there's a lot our audience can learn from each of you, especially because gene and cell therapy is an ever changing landscape. And so, before we get into some of the specifics about what each of your individual programs were like, I would like to jump into one of the questions from the audience, just as this, so we don't lose sight of that one. Particularly for Kim, can you give us a sense of how you went about finding more patients and conducting a natural history study? And then Michelle, after her response, you can always, and then please let us know from your perspective how it's advancing.
Absolutely. So we reached out to genetic testing companies to get our gene added to epilepsy panels. So because epilepsy begins so early in life and in our patient population, and because there are, there's different chunks of DNA that you can look at, right? So there's whole genome sequencing and then whole exome sequencing, and you can get down to these smaller and smaller bits of genetic material that you're looking at. And often the smaller the tests, like the fewer genes you're looking at, the more affordable. So for us, it made sense, especially at the time, to really push to have our gene included on epilepsy panels. The pricing of genetic testing has come down more. So I think there is, a shift in what is recommended, whole exome or whole genome even. But for us, getting on those panels was really key in those early years. In terms of starting a natural history study, I think that what Michelle touched on is really important, which is like if you can have a clinician who is championing your disease, like serving as your Sherpa across this process. And for her, that was Dr. Grinspan. For us, it was Brenda Porter out of Stanford. That was really key for us launching our natural history study. And we launched actually what ended up being a few studies. And that was partly because COVID happened right when we were launching our study. So we ended up launching a remote natural history study for a lot of our international patients with telehealth visits. We launched an in-person natural history study at 3 sites at Stanford, UT Southwestern, and Brown. And then we also did a medical records collection and analysis, and we did that through Citizen Health.
Fantastic. And then Michelle, from your perspective?
Yeah, so it's going to be a common theme that I skipped a lot of steps. So we were diagnosed, no patient registry, no natural history. We immediately went from bench science that had been done a hunch to manufacturing a drug to doing proof of concept and toxicity studies at the same time to dosing Elly. We now have come back full around. We've launched a patient registry with Dr. Grinspan, and we're launching a natural history study with Dr. Grinspan as well. I think, it's just the panel or Allison, when she spoke right before us, talked about the traditional pathway and the non-traditional pathway. And I think that's, a key point of what this panel is going to bring out. I had to go the non-traditional pathway. It's not necessarily the right pathway for everyone, but my daughter was so severe and was declining that was the route we had to go. But in some ways, to Kim's point, we were able to, we now have about 100 people in our patient registry. We know from kind of a Facebook group and other stuff, there's probably about 200 known patients, and then a lot of unknown patients. We also experience epilepsy pretty young in life. Not all of the kids, but a lot of the kids. We are not on the epilepsy panel yet, which is really how my diagnosis story took a little bit longer than it should have. We passed the epilepsy panel. We passed, various other panels. Thankfully, we got full genome sequencing, again, because of Dr. Grinspan and because we were in New York City. So we were able to get a diagnosis by 10 months of age, but many people are not. And so now as a foundation, we're circling back to do a lot of what Kim did to try to get onto the epilepsy panel, to try to make sure that we can make it more known. Our success so far in getting people to register has really just been the news of Elly being treated. And that has certainly motivated and encouraged people to go register.
Okay, thank you. And then Joe, did you have any comments, or how may you advise different patient groups, when they come to you in regards to should they start a natural history study, when should they do that, et cetera?
Yeah, I think sequentially, I think patient registry might be an easier lift first. And I think that's what you can use to start querying your patient community about, what sorts of modalities would they be interested in, and what symptoms of the disease are most problematic for them, or a pithy question is, if you could wave a magic wand and cure one symptom of the disease, what would it be? To sort of get that patient prioritization of the symptoms that they want to be addressed. And I think that can help inform drug developers or researchers when they're thinking about potential modalities or treatment paradigms. And then once that's underway, you definitely need the natural history study to understand, how does this disease progress? What are the measurable endpoints we might look at? And if you're going to go for a single arm trial, you're going to have to leverage that data set to augment only treating disease patients in that scenario. So it's a long-term process, and it can be very expensive to put up a natural history study on your own. So if you can find a researcher that's already interested in seeing patients and has some inclination towards this, perhaps the university can bear some of those costs. But that would be my advice. If you're starting from zero, start with the registry, and then find the clinicians that might be the sponsors, or the institutions that might be the sponsors for the natural history study, because you'll need it for whatever modality you eventually develop.
Yeah.
And I would just piggyback on that and say the FDA guidance, recently came out, and it really suggested that this natural history study data is valued and, will be helpful in terms of drug development and approvals.
Yeah,
especially
absolutely.
in the rare space.
Thing I would add, though, for very small organizations where money is really, really tight, the traditional natural history study, people have told me, we need $1 million. I'm like, well, I don't have $1 million for natural history. I do think we are pushing the industry a bit, and the industry is getting more creative in how to do it in a more affordable fashion, like potentially taking medical records that already exist, or doing virtual visits, or set up some sort of cross hybrid. And so, if you're sitting there in the early stages and people are telling you, okay, the first thing you need to do is natural history and you need $1 million for it, certainly we should be pushing that, because for these ultra rare diseases, we don't have that money. But I agree with you, Kim, the FDA is requiring it. It is something that we do need. And so trying to figure out a way to do that in a different fashion.
I also think natural history study and registry, and some of these terms, they are broad and have different meanings to different people. So you really need to think, I would say that what we did or are continuing to do is really a biomarker discovery study, so it is following the disease over time, but it's also specifically trying to pick out these biomarkers or surrogate endpoints, for clinical trials. So, just think about what what you're trying to do. Is it just to track the disease over time, or is it to look for these specific things for a future trial?
Yeah, absolutely. And as a researcher, I would say from my perspective, as we study these different rare disorders, try to develop therapeutic approaches, it's actually also incredibly helpful for the researchers to understand more about the patient population, if there could be biomarkers or outcome measurements that we can look at as we are doing our drug development studies, to then provide evidence to the FDA that you would expect potential benefit in those modalities. So thank you all for expanding upon those. So let's move now a little bit to some of your own personal trials and tribulations. So Kim, looking back at your transition from early research models to IND enabling work, what were the biggest scientific, regulatory, or operational roadblocks that you encountered that you hadn't anticipated from the start?
That's a big question. I'm going to chunk it a little bit.
Answer as you want to.
So the first part of the question was about the biggest scientific, regulatory, and operational roadblocks. Mm-hmm. For us, we touched on this a little bit in the intros, but because it was a newly discovered disorder, there weren't very many useful tools for scientists, and the tools that we did have weren't a perfect match for the disease. So, for instance, I don't use these terms to try to be offensive to the parents out there. I'm a parent too, but there was this fly model, and it was called the Indy fly, and Indy stands for I'm not dead yet. And it was a knockdown of our gene, and it should have been a good model. But the reason they called it the Indy fly is because it actually lived longer than the other flies, which is very different from what we see in the children, which is that they're very sick from the very beginning, right? So we have this problem of like what models are out there, and do they recapitulate the disease? Similarly, there is a knockout mouse, but it didn't have these severe seizures early on like we see in the kids. And so that was a real challenge for us. Do we create new mouse models? Do we move on to other model systems? What do we do there? And that's a question I think a lot of groups face, because our children are not mice. We've cured a lot of diseases in mice and not a lot of diseases in humans. And so I think that is an ongoing problem. In terms of regulatory roadblocks, I think that the field of gene therapy has evolved in good ways for the most part, but right along with us. So when we were doing our preclinical work, it really was like the mouse is the gold standard, and you have to go through non-human primates in order to get to to receive an IND and to do a first in human trial. And I think those are expensive steps, right? Like non-human primates are expensive models to work with for toxicity studies. And I think that now that AAV9 and some of these things have been used more, some of those steps are not necessarily required, but they were when we started. Like, I do not think that we could have changed the way that we have proceeded so far. And I think there's also a little bit of a question of like, what is the FDA going to require, and what are parents and families comfortable putting a medication in their child? Like, what have you done to prove that this is safe, right? So those are two slightly different questions that I would remind parents and who are going into drug development to think about. In terms of operational roadblocks, I think money is the biggest problem. I think it's really hard to find funding sources for these drugs, and there was a talk earlier, I can't remember, it was Steve, I think it was Steve Gray, who mentioned that like a lot of these, maybe it was Allison, I don't know, the morning's a blur, but it's been fantastic. They were talking about how, one of the things that often happened was families or foundations would start this research in an academic lab, and then they would de-risk that drug enough that a company would step in and take that program over and hopefully take it across the finish line. And that is largely needed because of the financials associated with drug development. There's also a lot of expertise that I think drug companies do have. So I'm not trying to minimize their role, but the financial piece is a big one, I think, for families. And so we had to, that is the path that we took with our gene therapy, was we did partner with an industry, with a company. And then when that company couldn't continue to support our drug, the program actually came back to us. So now we are the sponsor and the IND holder, and we are the one running the phase 1/2 clinical trial. Let's see, I think to kind of continue on that financial vein and an operational roadblock, we had to do a lot of things sequentially instead of concurrently. So I think Michelle will be a really great counterpart to this, because they were able to do, I think, a few things concurrently. So manufacture is a really expensive step here. And some groups will move forward with manufacturing before they've done a lot of the other preclinical work. And that is risky, because it means that, you could spend millions of dollars on manufacturing drug product and then not be able to use it if the rest of the preclinical information didn't look great. Let's see, that's the second part of the question. Oh, how did, let's see, internal capabilities, so those challenges revealed that we have.
I think you've answered most of the questions so far. There are some more that are within the chat that we'll get to, but I do want to pivot to Michelle, because we'll counterbalance what some of your perspectives have been with some of hers. So Michelle, from your experience, what are one or two inflection points where your efforts as a patient advocacy group were most meaningful in accelerating the progress towards a first-in-human gene therapy trial?
Sure. So I want to remind everyone that my program is very different. So Elly was very severe. We did an N-01, and we were able to fundraise relatively easily. So money was never an issue where we had to kind of raise the next tranche before we could move forward. We, we're very grateful and lucky that we were able to fundraise relatively quickly and easily. And we also were in New York City. So we had, access to RTW and Joe, who came alongside me, and taught me everything throughout the process. And we had other partners that we were able to, quickly identify because we sat in New York City. So I do want to kind of preface it with that. A couple of things that really helped us to move very quickly. One, we built a very lean, dedicated team. So our team was very small. It was me, Joe, Dr. Meyer, Dr. Grinspan, Andrea out of University of Missouri, and Susan DaCosta out of Jenison. That was basically it. And everyone had their role. We had our CMC expert, we had our preclinical expert, everyone had a purpose. And we assessed all the science that had been done, up to that point. We made strategic decisions in order to take multiple modalities as far as we could at the same time. And at some point when we hit roadblocks, we reconvened as a team and we decided to go all in on AAV9. The big thing we did, which Kim highlighted, that most people cannot do, is we started manufacturing before we knew if it was safe or if it would work. And that could have been an incredibly costly decision, $1 million decision. So we started manufacturing the plasmids at Charles River, and then we did the vector at Andelin. During the time period that the plasmids were being manufactured, we completed our proof of concept studies in Elly's cells. And thankfully it worked. It recovered her cells. We had a lot of very positive data. We had no, mouse model, mouse model, but we did induce a mouse model by injecting Elly's mutation and the AAV9 at the same time. When you just injected her mutation, the mice got very, very sick. And when you injected it with the gene therapy, they acted like wild type mice. So those two things we did very quickly, but they gave us enough of a feeling that we could move forward as parents from a proof of concept perspective. From a toxicity perspective, the vector manufacturing already began. So not just the plasmids, but the big, $1 million bill had already started, and we didn't know if it was safe. So this is, I think, where it was really scary. We did a toxicity study in 150 mice, and then we actually did 5 pigs. And Kim, I think bringing it back to something that you said, in our pre-IND meeting, we proposed this, and the FDA actually said, you don't need the pigs. You're using AAV9, you're using the same promoter that's already been in humans. We understand you have to go fast and there's cost considerations. You don't need the pigs. We did it anyways because of exactly what Kim was saying. I wanted to feel comfortable that I was putting something into my daughter that was safe. And I remember Dr. Meyer said to me, and she has a daughter the same age as mine, and I said, what would you do if your daughter had this? And she said, I would feel better with a couple pigs. So we did it not because the FDA required us to do it. We did it for kind of our own comfort. But, going back to the original question, a lean team, we moved really quickly. We were able to do steps kind of at the same time versus sequentially. And then the big thing is we acted as the sponsor. So in Allison's presentation before, she talked about the traditional versus non-traditional. We took a very non-traditional approach. So we hired everyone in our steps individually as CROs. We maintained full control. We wrote the IND. We submitted the IND. So Kim is now the sponsor and the IND holder, but she had to go through kind of the path to get there. I was able to learn from Kim and Amber Freed and all the people who came before me to skip that step. And that was very much a blessing because they did come before me and they were able to tell me to do it. So I would not have been in the position I was if I was diagnosed 10 years ago. I was able to really take the learnings from the parents ahead of me. And then the last thing is there was a lot of just divine intervention or luck or whatever you want to call it. We did hit roadblocks. We hit a lot of roadblocks, but for the most part, everything worked. And Elly was treated about a year ago. She's been seizure-free for 5 months, which is an incredible thing. And she is developing, we've stabilized the disease. So, in a lot of ways, stuff did work for us.
Fantastic. Thank you, Michelle. Joe, let's pivot to you a bit. So, we've heard 2 different pathways, for trying to get to the clinic where, as Michelle had said, if resources are not limited, what is possible? But, to Kim's point, not all families have those resources and are able to move at that speed or efficiency. So, if you were advising a newly formed patient advocacy group today that wants to catalyze the gene therapy program, what would you tell them to prioritize in the first 2 to 3 years? And are there mistakes they should avoid if they can?
Yeah, I think the advice is going to depend on the disease, the patient population. So, and that's what we do. We give very tailored and strategic advice based on all of these differences between the different diseases. But so, starting from a common point, I think you have to invest in understanding the disease itself. Usually, that starts with like a systematic research review or a literature review to understand what's out there, what models are out there, what cell-based assays are out there. If none exist, you've got to build those. So it's understanding the disease, understanding if the mechanism is known, what is the root cause of the pathology, and then you can start to get into ideas about like what would be potential therapeutics. But if you don't have that like mechanistic study really worked out, and Michelle had Kat Myers, who really knew IRF2BP very well, extraordinarily well. So that was so fortunate. You can't even start to do the drug development piece until you know some of that. So I think that's the first step. And then I think you do have to try to start some of these patient registries, query the patient population, understand what symptoms are of highest interest to them, what types of modalities are they willing to embrace based on the severity of the disease. Like not everyone's willing to get an AAV gene therapy injected into their brain if they don't have a really severe disorder. So I think you need to know that and query your patient population and understand what they're willing to do. Alana Newhouse from Foundation for Angelman Syndrome Therapeutics, in a recent panel sort of mentioned, a doctor said, "Oh, we can't pursue this drug because we'd have to put a port into your patients, and you wouldn't do that. We wouldn't pursue it." And she's like, "No, we would for some of these really severe Angelman syndrome kids." And so, like, you have to talk to the patients. You have to ask them questions, and I think that can inform the researchers. So that's the first place. Then validating that biology before jumping into the technology. And don't get married to a modality. Gene editing is neat and interesting, but it might not be the best thing for you to pursue. So understand the biology and select the modality that can really address the mechanism of the disease or the mutation type you have. Let the science guide you. Don't be swayed by like what's out there, most recent literature, most recent news articles and things like that. Really let the science guide you to the modality. And then to echo Michelle's point, assembling that team, you really have to assemble this team of champions, build your translational roadmap, think like a drug developer, think strategically. Don't just do science for the sake of research. This has to be research that you're funding that has intentionality behind it. It's either to fill in those gaps in your mechanistic data knowledge, your biology knowledge, or like what cell type, how much do I have to express to get a therapeutic effect? If those questions aren't answered, you got to answer them. And this should be strategic funding to get you all those answers, to get you to the modality that's optimal, and then develop that drug. Assembling this lean team of researchers, that's a must-have, I think, if you're going to really drive this forward with urgency. Your clinical expert, your disease mechanism expert, your regulatory expert, your modality expert. And they can't just be experts. They have to be experts that are driven by the mission. Like they have to be very invested in this too. They can't just be like popping in once in a while. They've got to be in it with you. So find those people, those champions, get them on board your team, and that's where you'll really move quickly. Other sort of pitfalls I see in this space, trying to do too much, that's not good. Like if you're trying to develop an AAV gene therapy, a gene editing modality, a small molecule repurposing, an enzyme replacement, all these approaches dilute your finite resources and your brain share of individuals that are working on it. So don't try to do everything. You can do a little of everything, but it has to be percentage-wise scaling. You should have the highest percent of effort focused on the modality that is most scientifically justified to treat your disorder and has like the confidence and rationale to back it up. So I think you have to really be strategic and prioritized in what you're doing in a percentage-wise manner. If funding is totally unlimited, sure, do it all, but that's not usually the case. And then, yeah, I just would encourage like reach out to more people in the rare disease community. I think that's one sort of superpower that this community shares. Everybody tells you how they did it and shares those tips and tools and tricks on how they did it. And they're really willing to help. We all want each other to succeed. So I think tap into that collective experience and expertise that exists among the rare disease ecosystem. There's power in that. I think like leveraging those examples and case studies that have come before you. Don't recreate the wheel. Like if somebody already made it, use their wheel, copy it. I think copying is the highest form of flattery. So like do that unabashedly and like ask for help. Someone might say like, hey, this sounds like what this organization did. Go talk to this person. So that's my recommendations. And then just to sum it up, I think the patient advocacy group, it's evolving over time. I think it was really just patient support, parent support. Now it's like doing the research, sponsoring the trials as these two foundations are doing, like potentially maybe taking it all the way. I hope they don't have to, but like they might, plan for the worst, but hope for the best. Like that's kind of the motto. So you have to be in this. And I think the goal of the patient advocacy group is to sort of de-risk everything that they can to the extent they need to in the hopes of enticing another partner to take it the rest of the way. For the rarest of rare diseases, that might never occur. For ones that have thousands of patients, potentially it may, but you kind of have to keep de-risking it up to the point where you could get that interest from the biotech industry or a biopharma partner that wants to pilot their platform technology. That's kind of the goal. So it's incremental and stepwise. And I think patient advocacy groups have to take the lead in the drug development piece in some of these really rare diseases. And I see this as like a trend that will only increase and is increasing because it's inevitable. It's not being addressed anywhere else. And once they see a few foundations do this, they know it's doable and they try to replicate it. So like it's thanks to pioneers like the TESS Research Foundation and Elly's team that it gives hope to a lot in the rare disease community that this is achievable and feasible.
Absolutely. So thank you so much for that, Joe. And you brought up, a very important point and one we've heard from both Kim and Michelle, that this trend of rare disease foundations that are now acting as the IND holders, the trial sponsors, and that's often because traditional academic or biotech sponsorship isn't viable, especially for the rare and ultra rare diseases. So, we do have a question from the audience, well, several questions from the audience around this topic. And one of that is a specific question regarding, is there an opportunity for patient group foundation to directly license a vector from an academic institution, even if the vector is already licensed to a larger biotech, who then happen to shelve that product? But they still hold the license. None of either of you or Joe, if you would feel comfortable speaking to that.
I don't know, Kim, this might be a better question for your org or.
I guess I need to know a little bit more about what the intended purpose, like if the drug, sometimes you can have compassionate use or expanded access, right? So if a company has a medication and you have a very sick patient, sometimes the license holder, like the drug company, the pharmaceutical company, will allow access to that medication in more of a compassionate use access. So that might be something to explore. I think that it really depends on the drug company. So sometimes a drug, like sometimes the contracting or the license will have clawback elements. Like some of it gets very legal and very, case to case in terms of what you can do if a program does get shelved. We've been so fortunate to have an industry partner who really wants to help our patient population and has been a very willing partner in terms of giving this program back to us so that we can move forward with the trial. So I think like a message to, pharmaceutical companies out there is like, there is a path forward for this handoff. And, that is something that I think also needs to be advertised. Like this is possible, this is happening. And I think it's such a wonderful thing when a drug doesn't get just shelved, when there is an opportunity for a nonprofit or some other entity to step in and try to help make these human clinical trials happen.
Yeah, absolutely. And then Michelle, I know you were able to avoid potentially having your program shelved as being the owner of it. Do you mind speaking a bit more behind what you had to do, I guess, to maintain this control and what it took?
Yeah. I mean, look, as I mentioned, I learned from parents who came before me. When I was first diagnosed in the first couple of months, all I did is I talked to anyone and everyone who would talk to me. And there were consistent themes about, make sure you maintain control, try to maintain IP, try to, keep as much as you possibly can. And so I was able to learn from all the people that came before me. And in a lot of ways, I was able to do that. And also, I didn't let contracting, I didn't let IP, I didn't let legal agreements hold me up from treating my child either. And so, there's a lot of ways where I was able to put joint IP in, or some sort of we'll deal with this later, language, like if it ever is a commercial product, if it ever, because for us, I mean, for the ultra rare, it most likely never will be, right? And so I think I tried to, maintain as much control as I could, and also not let it hold up treating Elly and getting to the clinic.
Absolutely. And then, Joe, would you have anything to add to this? Because I think,
Yeah.
the intellectual property part, trying to maintain ownership can be very scary for parents who are not in that type of field or environment. And then, is there then potential downfalls if it is maintained by the patient advocacy group in terms of later development or bringing on biotechs at a later point?
Yeah, I always advise for ownership and usage rights, like for research purposes at a minimum. I think retaining ownership and control over the IP also enables swift decision making. And like if you have to negotiate with a CRO or a CDMO to manufacture the drug, it's just your team deciding it. You don't have to go through the university or go through like whoever else is the IP holder. So I think it can lend some efficiency and speed to the overall effort if foundations do own their own IP. But then again, that also puts a lot of burden on them in terms of patent filing, patent writing, getting attorneys. There are a lot of good pro bono attorneys in the rare disease space. So like some of them can be of assistance, but if you're actually filing a patent, defending claims, that's a bit more than what I think a pro bono attorney might be up for. But I would definitely leverage these pro bono attorneys to review your contracts and your clauses and your terms that you have in there to make sure that there are favorable terms for the foundation. I would never advise to just do like unrestricted gifts or donations. I would always do this as an agreement with terms. They'll vary according to the disease and what the tolerance is for each foundation. But at a minimum, I would say like anything you're funding, you should be able to use that data, use that reagent, use that mouse model, use that cell line, and share it with whoever you want that can do the next piece of the work that needs to get done. This shouldn't be locked behind individual institutions. Some of this work should be stuck. It should be sort of fungible, and you should be able to move it around to whoever can get the next piece of that stuff done. So that's what I advocate for with the foundations that we talk to. I think a lot of these foundations have different contract terms and clauses, and they're willing to share those from their agreements. And then you can get that overall contract vetted by a pro bono attorney. So like that's sort of what I advise in this space. And then just to follow up on the compassionate use, I mean, one of the scenarios that I see sometimes is there could be an existing drug that's still in clinical trials by a large pharmaceutical company, and you might have some evidence that that drug could be usable in a very rare disease. The common prevailing view in industry is that like they should never do that because that can imperil the development path for the bigger, larger, more profitable disease, and that's what I hear. But I think there are some changes emerging under EAPs and compassionate use protocols where there could be some incentives to testing some of these drugs that are still in investigational pathways right now, and leveraging some of the responses that are collected in those compassionate use trial to support the regulatory approval. And oftentimes, like in these rarer diseases where the mechanism is so well defined, the probability of the drug working might be higher in that extremely rare disease than in the bigger disease. So I think industry partners need to reevaluate how they look at EAPs and compassionate use protocols, and not just sort of say like, oh, it could impact this bigger trial we have going on. So I'd love industry partners to sort of recalibrate their views on usage of these types of investigational drugs.
Great. And then, so we've heard good, fantastic examples. If you're starting out, what would be ideal? Kim provided an example, if your vector is licensed by a company who shelves it, is there possibility of getting ownership? And in her case, it was feasible through I wouldn't say easy, but feasible through conversations, with that partner. But, our audience does ask, maybe Joe, you would know, are there other options if a vector has been licensed to a larger biotech and they are not necessarily giving it up in terms of establishing ownership?
Yeah, I think I'll just echo Kim's comments. Like if it's licensed for a specific indication that is different from your disease that you want to treat it in, you can apply for a separate license depending on the terms. It might be just licensed to the overall technology, in which case I think it would be, you'd have to negotiate with the biotech company that holds that license. But if there's wiggle room to get an additional license from the original creator, the patent holder, specifically for your disease indication that is differentiated enough from what the biotech company has licensed it for, there might be room for that. So I think it's worth investigating and exploring. But I can't sort of comment too detailed on that without knowing more details about the specific disease.
Right. And I think it's an ever-changing landscape again, and depending on the partner, this could look very different. So thank you for that. So let's switch a bit maybe to some of our safety questions that I see in our chat. So some of them are, Michelle, why did you opt for pigs rather than non-human primates? And then to piggyback on that, did you learn anything from the pigs that you didn't already know?
Yeah, so we did mini pigs at the University of Missouri. We picked it because they have a mini pig farm, and they're very, very good in that model. And University of Missouri is the organization that we outsourced our proof of concept and our toxicity work to. So it presented an opportunity to do a larger animal, but in a less expensive way versus a monkey or something like that. And, Dr. Meyer felt she has used mini pigs a lot. She felt very comfortable that it would provide us with better, stronger comfort regarding the safety versus just the mice. And so that's what we did. Did it provide anything further? I mean, the pigs were great. They were safe. They lived. That everything was very safe with our product, which is great. I think one of the questions I often get is, after all of it, the day you're sitting in there in the hospital when you're about to dose your child, how did you feel? And I remember thinking, and very few people will ever have to feel this, but many who are listening, who are striving to do what we're doing will. I remember sitting there and thinking, okay, I might have created something that saves my daughter's life or that kills her. And you're sitting there and I hand my baby over, and I remember being comforted that we did the pigs. So what did it teach? What did it tell me? Well, the pigs were fine. But in that moment, when you're handing over your child, like having just the comfort to go back to the science, to go back to the studies that you did. And then ultimately, at some point, you just have to rely on faith. But having the science and having the pigs did give me that comfort on that day.
Fantastic. And then, for Kim, from your program, although with the non-human primates, part of that was FDA responses as to what was suggested, did it also, from a scientific standpoint, make some sense or provide comfort, I guess, to your program to test it in the non-human primate versus maybe something like a pig?
Yeah, I mean, I love Michelle's answer to that, testing these, we're talking about our own children for a lot of these diseases and drugs. And so it really does, the science is like reassuring, I think. But specifically with non-human primates, I think dosing is a big deal. So, what we're talking about is a gene replacement therapy. And Dr. Gray talked about how right now we can't redose these, right? Like we are exposing, we are using AAV9, and that right now that is, can't be repeated because your body is then exposed to that virus, and it's not possible to redose. So for us, we wanted to make sure that we were going in with a dose that was like the highest safe dose, so that we could hopefully maximize efficacy for families, even in patient number 1, right? Like some of these trials have dose escalation where, non-human primates are maybe used to try out different doses until you get to a toxic point, so that you can figure out, what that dose in humans might be. So I think it's really helpful in dosing, just because non-human primates are large, like, humans, like our children.
And Rachel, one thing I want to add quickly that actually did help us speed up, we used the clinical material in the pigs. And so we did not have to do batch comparison between the research grade and the clinical material, which for some organizations can be a huge nightmare if you figure out that your clinical material is much more potent than your research material. So I do think our FDA path was faster because we used the pigs, because we were able to use the clinical material at the exact dose, when you did the dose calculations as what Elly was getting. And so we were able to say these pigs got the exact same treatment that Elly is going to get. And that significantly helped our regulatory pathway as well.
Thank you so much for that point. And probably just the last thing I would add is, for the audience, is also to think about the science of the disease of the gene you're delivering, because sometimes a gene is actually not very highly conserved in a rodent compared to a human, or a different type of animal compared to a human. And so if there are potential toxicity concerns, you may not be able to fully test it in an animal model where it's quite different. And so that is also something I would just pose for our audience to think about, is the science behind it and what also makes sense for your gene, for how large of an animal you may want to go and how comparable it is to your disease. So, Joe, there is one more question in the chat that we don't have time to get to. So if you're able to put anything into the chat to let the group know, you can do it through the all chat. We can see if there's any advice you can give on that. But at this time, I'd really love for each of our panelists to give us kind of your wrap-up summary of our discussion. And Kim, we'll start with you.
I know we only have a few minutes left, so I think I'd boil it down to like 2 main points, one of which I haven't touched on, but I think Michelle did. This idea of lean teams, like building your team is really important. And I know that I talked about how we had a clinician who helped us launch our natural history study, but I also just wanted to give a shout out to our scientific director, Dr. Tanya Brown, because having an in-house scientific director is a game changer for us. And that was made possible by a grant from the Chan Zuckerberg Initiative, which was a capacity building grant. I think that grants like that are really powerful for nonprofit organizations, and having that expertise on your lean team is amazing. And the second thing I would point out is that I think that none of this is easy, but there is something of a path for N of 1. And there is something of a path for N of rare, but just barely rare, right? Where you have a commercially viable market. I think there is not a clear path still for the ultra rare communities. And so I would just continue. I was so excited to hear Dr. Flott talk about like this clearinghouse and marketplace that ASGCT is partnering on. I mean, I think those type of financial changes are really needed in order to make a path for the N of rare, N of ultra rare.
Thank you, Kim. And then Joe, final thoughts?
Yeah, I think patient advocates, patient advocacy groups, foundations, they're innovating new paths for therapeutic development. This is a trend that I see increasing. I mean, Fox G1, TESS Research Foundation, Elly's team, they're all going this very scary route. It's unfair that they have to do this at all, but I'm hopeful that as more do this, they share those learnings back and it becomes easier. I also hope that regulatory pathways adapt to remove or shift some of this unfair burden that we've placed on rare disease parents and foundations, so that it does shift back to the drug development community. Because I think this is kind of crazy that Michelle and Kim have to do what they're doing, but they're doing it. I just hope that the system recognizes the inequity here and adapts so that we can let parents and patients be parents and patients, and let the drug developers take on some of this aspect. But until there are lots of changes that occur in the drug development ecosystem, I think this is going to become an increasing trend that we see. So I'm just going to try and do whatever I can to help these parents move these things forward. And I think everyone else in the community is supportive too.
Very well said. Thank you. And then Michelle, your final thoughts?
Yeah, I mean, I think to just echo everyone, when we were first diagnosed, we talked to many, many people, and most of them told me it was going to take 5 to 10 years and I needed $5 to $10 million. And we did it in a year for under $2 million. And I think these stories need to be told, and we need to push the envelope, particularly as parents, because even a year might be too slow for some of our kids. We need to move quicker. And so for those of you who are listening, your path is not going to be the same as mine or as Kim's, but we are all part of this relay race where we are pushing the science forward. We are pushing these developments forward. We're pushing gene therapy forward. And we're handing off the baton to each other. And so because the parents came before me, I was able to do what I did. I hope that because of what I did, parents can do something even better than what I did. And so really coming together as a community, really recognizing that every single thing we're doing is pushing the industry forward and truly changing the world, I think is what can keep us going. And you might not get to the end, you might not get to the clinic, but whatever it is you did mattered, and it moved the race forward.
Thank you so much, Michelle. That's fantastic. And so as we wrap up for today, I want to pause and recognize the collaboration and openness everyone brought to the conversation. We covered a lot of ground, from exciting new programs emerging in the CGT field to various treatment approaches and safety concerns, then how researchers pursue specific treatments, and by finally examining the critical role patient advocacy groups play through firsthand perspectives from those leading and supporting CGT drug development. The questions, perspectives, and dialogue today are what make this summit so impactful. I want to thank each of our speakers, panelists, and you, the audience, for your time, energy, and thoughtful participation. Your insights truly made this discussion meaningful.